Hello everyone, my name is Dominic Craske. I'm a final year PhD student here at the University of Manchester, and the title of my presentation today is Atomic Force Microscopy, a tool to study early stage corrosion. So before I start, I'd just like to introduce people who are involved in this project. Rob Lindsay, he is my supervisor. Chris Murin, he has given me technical help and advice when it comes to using AFM. And Hannah Woods, who is the technician who's in charge of the AFM I use in NGI. So in this talk, I'll talk about the motivation and the goals behind this project, as well as going through some basic theory of AFM. I'll talk through our experimental setup and then through some baseline experiments that we performed before getting to some in situ AFM results, which I've got. So firstly, our motivation. In industry, carbon steel is extensively used in service components such as a pipeline in the oil and gas industry, for example. Carbon steel is used because of its comparatively cheap price compared to something like stainless steel, and also its appropriate mechanical properties for the application. Despite this, however, it is unfortunately susceptible to corrosive attack by weak acids or in corrosive environments. So corrosion caused by weak acids can be detrimental to the integrity of the service component. Alternatively, in some cases, it can actually help to protect the steel from further corrosion by forming a corrosion scale or a protective layer on top. So in these images at the bottom of the slide, the image on the right, this one, is that the right or the left? Yeah, that's the right. Is a piece of carbon steel which has been polished to 1 micron, which has suffered corrosive attack by acetic acid. The other image is SEM image, these are both SEM images, of an iron carbonate scale which is formed on a piece of high purity iron that's been exposed to carbonic acid. So when studying corrosion, there is a need for in situ techniques. Taking these metals out of the environment to measure them can cause changes in characteristics of the surface of the metal, such as you might get an oxide film form that wasn't initially there. This is a particularly large problem when you're looking at the early stages of corrosion, because small features that you might want to see in a microscope, for example, might be obscured by new things that have happened after you've taken it from the environment. So the goals of this project are to implement AFM for imaging corrosion as it happens, specifically the early stages, and more specifically looking at acetic acid corrosion of carbon steel. And we'll explore how the different phases in the steel act and how to the corrosive medium, and also how roughening changes over time. We hope in the future to then apply this methodology to carbonic acid systems. However, these are much more experimentally complicated because it requires a low oxygen environment. This is to see early stages of iron carbonate formation. I'll come back to this work plan in a future slide. So now moving on to the theory. So in very basic terms, atomic force microscopy can be thought of an imaging equivalent to a vinyl record player. So we have a probe, I'll try and point my laser at it, which is attached to a cantilever. The probe is moved across the surface of the sample. There's a laser, showing it, which is shined on the back of the cantilever and then reflected onto a photodetector. So this tracks the movements of the cantilever as this probe moves along the surface. And on the right side, I've shown a photograph of the AFM that I use. This is the JPK Nano Wizard 4. This is a top-down tip scanning atomic force microscope. And this is encased, I don't know if you can see it in this image, but in a soundproof cupboard, there's some soundproofing there, and is on an isolation table, and this is to help with stopping external noises and vibrations causing problems when imaging. Sorry. So there's the three most common imaging modes thought of when we talk about AFM are contact mode, tapping mode, and force curves mode. Contact mode, the probe is in constant contact with the sample as it moves along the surface, and it is imaged as it goes. In tapping mode, the probe is oscillated over the sample, and the height is measured when the tip comes into contact with the surface. In force curves mode, the probe is pushed into the sample until a set force is reached, and the height is then measured. So in my work, I use force curves mode. This is purely because this is the best, we've got the best image quality and consistency in liquid systems than when we tried tapping mode. So why do we want to use AFM? So AFM is not, unlike other high resolution imaging techniques, is not restricted by environment, such as SEM, which needs a vacuum chamber. AFM can be performed in air, in inert gas, it can be even performed in vacuum, where another mode called non-contact mode can be performed, which can give near atomic resolution, or if not, atomic resolution imaging. And because of these reasons, and the fact that we can image in air, it makes it relatively easy compared to other techniques to image in liquid. Additionally, added to this, people who produce AFM, or the companies that produce atomic force microscopes, often produce stock add-ons such as electrochemical cells or heating stages, which allow other techniques to be used at the same time. So we can measure corrosion rate, or we can do heating experiments. And this means that we don't have to produce our own bespoke cells for these sorts of experiments. So just a few pros and cons. I've already mentioned in the pros that the specialized environment and liquid imaging is possible. But the other thing is we get a true 3D surface profile on our image with which we can actually measure the height. This is in contrast to something like SEM, which, although it appears 3D, there is no way to measure the vertical axis. Apart from this, added to this, it has very good lateral resolution, and this is limited by the tip size that you use of the probe, which tends to be around 10 to 20 nanometers at the very bottom. It also, as I said, has good vertical resolution. Problems with AFM are things such as that it is more prone to artifacts than other type of imaging techniques, and this is inherent to the technique. This could be something like your probe, there's a problem with your probe. It could be blunting, breaking, splitting, or picking up dirt somewhere on your sample, causing things in the image that aren't real. This is up to the user to be able to see what is and what isn't real, and this is not necessarily that straightforward. Acquisition times for imaging are in the ballpark of several minutes at a minimum. This is a particularly big problem for us because in force curves mode, this takes even longer because it takes pixel by pixel, whereas something like tapping mode takes line by line. The field of view on the specific AFM I use, the maximum size I can image is a 100 by 100 micron area. Now, in my work, this is not a problem because I want to see very small features coming from early stage corrosion. So, not necessarily an issue for me. Also, it can damage the sample. This is, I believe, a problem more associated with contact mode, and it's not an issue that I've ever seen. So the work plan. Start with dry imaging as a baseline. You need to be able to dry image before we can move on to more complex systems. So this is a piece of carbon steel which has been polished to 1 micron. On the image, I show a scale bar on the right. This is the vertical scale in nanometers. The image shows polishing scratches from the sample preparation, and these are quite shallow. We're talking 5, 10, maybe 20 nanometers deep. But this gives you a good idea of what we can see on a polished piece of steel. By eye, this steel is, or this sample is a mirror finish. After this, we need to start being able to image in liquid. So here I show another AFM image, this time imaged in deionized water, carbon steel again, and we see very similar image quality. This shows that in AFM, we don't actually sacrifice image quality if we go into liquids, which is another advantage. So from here, we need to start looking at in situ corrosion, and the system we use is acetic acid, and this is the results I'll talk about today. So now onto our experimental setup. This is the cell that I use. This is a stock electrochemical cell from JPK. The cell is here. This sits on top of the sample, which is in the middle here, and is sealed against it with an O-ring. this gives a liquid tight seal. Liquid inlets and liquid outlets allow liquid to be injected or removed from this sample. The cell can hold about 1.5 millilitres at a max. On the back of the cell, there is a spring-loaded gold contact which pushes into the sample. This allows it to be used as a working electrode, and a platinum counter electrode is coiled around the inside of the cell. There is a slot at the top for a dry ref micro reference electrode, so we can do full electrode we can do 3 electrode setup experiments with this. Our substrate is mild steel 1010, polished to 1 micron, and our solution is a 0.09 molar acetic acid solution, approximately 1.2 millilitres. So you may notice that this is quite a small volume, so we thought it necessary to do some experiments to compare to larger volume sort of solution. So for this experiment, we just used a 3 electrode setup, same substrate, same solution, and corrosion rate, LPR was measured and corrosion rate calculated from that. And this is our setup on the right. It's just a glass cell, liquid in it, electrodes in, nothing special. So the graph on the left shows the average corrosion rate over 3 runs in the 1 litre of solution, so the larger volume cell. The graph on the right shows the corrosion rate in the EC cell of the AFM. And in both cases, it seems to stabilise at around 0.2 millimetres per year. So we believe, at least at these timescales, that the corrosion kinetics are roughly the same. So these are comparable systems, at least at these timescales. I'd also like to point out that the different end times for the AFM runs, we run these samples until the AFM probe breaks. So that is why each of these have a different endpoint. So, getting on to the actual in situ results. So, this was a piece of carbon steel. This was immersed in acetic acid, and this is after 195 minutes of immersion. So, with this image, we can see all of this looks fairly similar, but there's this feature in the top right of this image. I can't tell my right and left at the moment. So this feature comes out quite. It's your views are right from mine. That's the problem. This feature sticks out quite a lot. In initial experiments, we thought these were artifacts because these were much smaller generally. But seeing such a large one, we thought maybe there is something more to this. So by changing the contrast on the image or adjusting the Z scale to highlight this section, we can see that it remains relatively flat, whilst also containing what appears to be polishing marks left over from the sample prep. So a little bit about the carbon steel. It has got 2 phases in it. It's got cementite, which is Fe3C, or iron carbide, which is the minor component, and ferrite, which is just what we think of as iron. So we wondered if this was something to do with this. So what we did was we took this same sample, we dried it, after we washed it, we dried it very quickly, put it into a vacuum desiccator, stored it there until we could take it to an SEM. The middle image shows an SEM image of exactly the same area shown in the AFM image. And what we can see is that this thing is indeed real. Unfortunately, in the process, it did get a bit of muck next to it, but that's not really an issue. We then performed electron backscatter diffraction, or more commonly known as EBSD, on this sample, and this is shown on the right. So this is a phase map. Red is used to represent ferrite, blue is used to represent cementite. And the yellow square on this is the same area that is shown in these 2 images. And this did indeed confirm what we thought, that the cementite was these features. And this is sorry, I'll move on. So we started looking at these cementite features. Again, this is a different run of the same experiment, and we have images after 94, 137, and 197 minutes. Now, this line on here is where we extracted a line profile from the sample, going over the cementite to try and explore it a bit more. We then normalized the height data so it all matched with the cementite. So the reason we did this is that we noticed that the cementite seems to be getting brighter from image to image. And this suggests it's increasing in height in the Z scale, whereas the ferrite is getting darker, so it is reducing in the Z scale. So what this quite nicely illustrates is that the cementite is probably actually staying in exactly the same place, not corroding at all. And this is what we really expect, because cementite doesn't corrode. Whilst either side of it, we are getting corrosion of the ferrite. So I'll now move on to roughness data. So another thing we can look at with the AFM is roughness data. Is that going to play if I click that? Yeah. So this video just shows, it's the same experiment as the last one. And it's just a run through of what we did with the experiment. The whole experiment, every image that was done in this. And we can extract roughness data in the image processing software, and then we can plot the roughness data over time to check how it's evolving. We see that the roughness seems to be increasing linearly with time, although we believe that after some period this will probably plateau off. So it'll be linear, nice straight line, and then it'll flatten somewhere after maybe another hour or two. We hope to show this in future experiments, but this isn't actually that easy to do. So this will be, we're trying. What I didn't mention is this is 3 separate runs of this experiment, all starting from the beginning. So why is there these graphs are showing quite different profiles for roughness over time, and this is despite the fact that the electrochemistry or the corrosion rate data suggests that we should be seeing the same sort of thing. So if the corrosion rate is similar, we expect to see the same sort of thing at the surface, right? So some of you may have noticed earlier in this slide, which I showed you, that each side of the graph is not at the same level. And some of you may have even noticed that this cementite appears to be lying on a grain boundary. So this got us thinking that it's probably to do with the microstructure that we're seeing this different roughness between different experiments. So here is another video of the same experiment. And this again just shows the roughness plot over time. So we're sort of seeing it is kind of an etching effect, but we're revealing the microstructure, we're revealing the grain boundaries as we're doing these experiments. So what I can do is I can take these images and I can mask off specific areas. I've marked off the grains, I've numbered them, or what I think are the grains, numbered them, and then plotted roughness for just these specific grains. And indeed, what we see is quite a distinct difference in roughness between the 2 grains. Additionally, I don't know if you're going to be able to see this or not, the features seem to be going in different directions. So if we look at grain 8 and we compare that to grain 2, so grain 8 looks like it's sort of roughening sort of to the right, whereas grain 2 looks like it's sort of roughening up and slightly to the left. I don't know if you guys can see that, but that is how we see it. So we believe that these effects are to do with the orientation of the grain. So the grain that's exposed may have more or less active sites for corrosion to occur, depending on which crystal facet is exposed. And we hope to explore this in the future, but we haven't got there yet, by looking more deeply at the EBSD data and trying to compare the roughness of grains to known orientations. So I'll move on to our summary and our future work. So we have shown that AFM is an appropriate technique for looking at corrosion as it happens. We have also observed that between different experiments, different roughening rates of the entire sampled area, and we've determined that this is due to the grains that I touched down on and then subsequently image. We've also done a bit of correlation between AFM images and EBSD data to look at how the phases to distinguish the phases and work out what they're doing differently. So going forward, we want to try and do that thing where we image till we get a plateau in the roughness that we believe is going to happen, And then we want to also carry on exploring the barrier of EBSD. And I've got the orientation map from the same EBSD which we did we distinguished the cementite and the ferrite on. So we also are going to repeat these experiments in carbonic acid, and this is to image early stage formation of iron carbonate scales. So finally, I'd like to thank BP for funding and technical support through the BP ICANN, Prosperity Partnership through the EPSRC, and the Materials for Demanding Environment CDT. Thank you.
Thank you, Dominic, for a nice talk, presented on time, which is better than most. Anyway, do we have any questions? We started a little late, so I'm prepared to run a little into the lunch break. Yes.
Thank you, Dominic, for your presentation. All the images that you showed are topographic, topographic, yes, the topography of the.
Yes, they are.
But with AFM, you can also obtain potential maps using the scanning Kelvin probe atomic force microscopy. Have you thought about using this technique? This is quite interesting for corrosion studies because it allows you obtaining maps where you will be able to identify the anodic and cathodic sites. So,
this isn't something I know much about, I say anything about. So I have heard of the technique. I don't know what it does. So it would be quite interesting because I guess we could see if the cementite in our samples is acting as the cathode for, and the ferrite is acting, well, obviously it is the anode, but we could see possibly that. But no, this isn't something I've done. It's not something I know anything about. So.
Well, thank you.
Cheers.
Hi, thank you for that. I had two quick comments. One was the same as Beatrice, which is you should really do some scanning Kelvin probe in the AFM. And don't be frightened of it. It's simple. If that machine you have does phase contrast, you can run that. Go read Martin Stratman's papers to work out what it's telling you. And it will take away all these questions about what you're looking at, right, chemically in the system. I guess I wanted to understand a little bit about how you're doing the correlation to the EBSD. So there are lots of ways that people try to put fiducial markers onto surfaces. One way I've seen people do it is right at the end of an AFM experiment, you crash your tip, right, on the surface, and then you can find that your EBSD maps are good. So there's that. That was one question. Then just very quickly, the people that are working in Bristol on the really high-speed AFM now, how does your work compare to what they're doing? And is there any advantage for you doing it a little bit slower? I mean, your process is slower, I think, compared to something they're looking at. But where does your work fit into, I guess, that scheme of people pushing for speed, maybe versus the other types of information you're getting? As for the fiducials, we initially started off trying to use micro indentation to give the same area between them. But actually, we found that the mechanical deformation caused by the fiducial was causing quite a lot of problems. So what I decided to do is I started scratching the samples with a scalpel. And I do a cross pattern, and then I do from corner to corner, and then I do another one going down. And this, hang on, is that right? Basically, we make a triangle with scalpel marks in the middle, as small as I can by eye. And that's between the grinding and polishing steps. So we then polish the sample afterwards, which reduces the deformation, the mechanical deformation of the sample slightly, which helps a bit. It's still quite tricky because I don't have the AFM that we use doesn't have a good camera. So when we're trying to touch down on a specific point, I can get into that triangle, but I don't necessarily know where in that triangle I am. So taking it then to the SEM can be a bit time consuming. I think it took me about 3 or 4 hours to find that specific place. Is it after you've done the experiment, are you able to get good EBSD data then?
Yes, so that EBSD map was taken after it was corroded.
I would be tempted to crush your tip right at the end. You know, you're going to change it anyway for the next experiment. Go to somewhere slightly away from the position of interest, and you'll find it straight away. You'll find it in the SEM.
Yeah, that is probably true.
It's a bit brute force, but
don't want to risk also possibly damaging the AFM. I don't know how hard it is. I don't want to do something like that. It's got a quartz crystal on it that I don't want to scratch.
Have you got access to the force distance curve?
Data, the
Yeah,
force
so
it's not particularly useful.
but it is if you want to slightly crash your tip, right? Because you know exactly what force will hit the surface.
Yeah, that's
We can
true.
maybe talk about it later.
Also, you mentioned the high-speed AFM. This would be better if we could do this at high speed, but one thing to that is the tip probably wouldn't last as long if we were doing, say, video rate scanning, but I guess we could solve that by only imaging once every half an hour still. So these images do take 20 minutes to take. It would be better to have a high-speed image. I don't know the specific work that you're talking about, so I can't comment specifically on it. But yes, it probably would be better, but we don't have that capability here in Manchester, unfortunately.
Okay, if you talk to me afterwards, I'll give you the reference to the people.
Brilliant, thank you.